I saw your look.
I thought that long. No. I don't know how long it's been on.
I don't know. It's for undergraduate, so applying for one job at U of M in their neuroscience department. And I just start CBS.
Is that ACD?
Yes.
As we make your margins bigger, sure. Get longer. Oh, they look a little narrow.
Oh, should I make cards like?
But so long as it does what you've done, you know, it's not like someone is counting the pages and marking down exactly how many it is. It's just you want to be exhausted about everything you actually have done. Why is the display not turning on?
Is that note on the thing about the projector? Is it behind
There's a note?
on the board?
Ah, okay. Yes, there is a note. All right. Okay. In that case, you might be able to see a little better from that little on the end. If we're looking at the slides. I think that will circle us around this projector. So you can see from there. Okay. All right. Sorry for the quick change of plans, both last night and today, everyone, but I do hope we'll be able to get outside soon. Just today didn't quite work out the way we planned. So instead, we're going to get started thinking a little bit about the ferns. So our next lab will be looking at ferns. So this is a little bit of a reminder about our phylogeny of plants overall. So when we think about the origin of plants, the origin of land plants, we have them roughly branching off from algae, although we saw on that practice worksheet that if we include all our algae, the situation gets a little bit more complicated. Then we have our first branch. So our first branch in the tree of land plants are the bryophytes, or the mosses. These are not vascular plants, so they don't have xylem and phloem. They don't have vasculature. They don't have vascular structures. So if you see moss on the ground, it's not moving these fluids around using vasculature. So we're not going to focus on the mosses in this class, although if we have time at the end and anybody's super interested in moss, you can take a look. That speaker we heard, the first one, Dr. Kimmerer, I think is an expert in mosses. So if you looked into any of her other podcasts or anything, you may have heard her speak about moss rather at length. Okay. So what we are talking about today then is our first branch of vascular plants. So these are what we've termed our ferns and fern allies previously. So our seedless vascular plants. So they're going to have xylem and phloem, they're going to have vasculature, but they're not going to have seeds. They're going to reproduce using spores. So that's how we group these together. Ferns. Our reference chapter for ferns is chapter 7. So as you're looking through later, there'll be a couple questions at the end of this set of lectures. I'm going to ask you to briefly read from your textbook as I say, studying for our lecture quiz. Probably you'll see one of those lecture questions based on the reading appearing on our quiz. I think that's a fair way to assess that. Okay. So today we're thinking about ferns and fern kin or fern allies, which are otherwise known as pteridophytes. So pteridophytes are vascular plants that don't have seeds. So pteridophytes can be broken into two groups, roughly, the lycophytes, or the lycopod group, and the monophytes, or the fern group. The lycophytes branch off first, and the monophytes are going to have further divisions. So if we think about our divisions overall, we're going to put our lycophytes in their own group, so that's going to appear as number 1 in a second. But then as we divide the monophytes, we're going to divide them into the whisk ferns, or Xylophyta, the horsetails, which are Macrophyllophyta, lycophytes, and the true ferns, or the Polypodiophyta. We're going to have club moss, our whisk ferns, our horsetails, and our true ferns. If you reference other phylogenies or other texts, as we've talked about with classification and systematics, this is the kind of thing that scientists argue back and forth about and rearrange pretty commonly. So other sources will use slightly different arrangements. I kind of recommend actually for this unit focusing on those English names, because those tend to hold fast a little more than these specific scientific names, because they keep renaming some of these groups. Okay. So here we might see a map, and you can see already that the scientific names are slightly different here. But one thing we can see is that those lycophytes are going to be one of our basal group here. So they're kind of our out group within the rest of this fern and fern allies grouping. Jumping up here to the top, we can see our true ferns branching off, and we can see that in this phylogeny, we have our true ferns as a sister group to our horsetails. That's here, and we can see their common ancestor here. And then we can see our whisk ferns having a more recent common ancestor with the true ferns and the horsetails, and then a farther common ancestor with the lycophytes. So your book has a table like this, dividing these things into a lot more classes and listing the orders that go with them. So this is in the slides, but just for reference, if you're looking at that, we're considering this first division, our lycophytes, or our Lycopodiophyta. Then this class B is our Xylophyta club. So that's going to be these two orders here. Our class C is the Microphyllophyta. And then everything from D down is going to be in that true fern group. So we'll see some examples during the lab of some of these orders on Thursday. So most ferns are perennial. So if you see a patch of ferns in the woods in the spring and the summer, bless you, you can expect that it will be popping up there again in the next year. And that can be helpful if you are looking to, for example, collect new growing ferns. So they're usually found in moist, shady terrestrial habitats. So wet woods, a rainforest like this one would be a great place for a fern, but we will see them in some other places as well. For example, some ferns are aquatic. So some ferns live in water, even though we are thinking about ferns as one of our groups of land plants. Some ferns are epiphytes, and an epiphyte is like a plant that doesn't grow on the ground. It grows on another plant or some other type of material. So it's not drawing nutrition from the soil. It's drawing it basically either from the other plant or from kind of a hollow in another plant. So anything that's kind of may be an epiphyte. A couple of ferns do live in arid places. And there's a cool fern called the resurrection fern. I haven't looked totally into its life cycle, but just from the name, I think guess what it does, and maybe by Thursday I will know a little bit more about the resurrection fern. There are a lot of species of ferns. About 2/3 of the 11,000 that we've named so far are found in tropical regions. And ferns are actually contribute a lot to coal, so coal deposits, and historically during previous eras, ferns have been much more common. So in the past, they were much larger and more widespread. Ferns have very variable morphology. Some are very small. One called Salvinia is small and aquatic, but they can also be truly enormous. There's a 75-foot tall one, Cyathea, and the fronds are 23 feet in length. So like the fern leaves, that leaf structure is called a frond when we're talking about ferns, 23 feet long. And so super, super variable in what they're going to look like. I'll just pull some pictures of ferns. So here we have some examples of aquatic ferns. So these are aquarium plants in this case. Here we have some epiphytic ferns. So here you can kind of see what I mean about what an epiphyte is. So here we see the tree, that's called a ficus that this fern is growing on. And we can see it's kind of grabbing on into the trunk. Or here we can see ferns hanging down, kind of out of this mossy structure that would be on a trunk as well. And here are a couple pictures of tree ferns. I believe these are from Australia. And you can see they have a trunk-like structure, and then almost kind of like a palm tree looking. These fern fronds branching out from the trunk. Oh, and some desert ferns. They do exist, but they are few and far between. So most ferns do have roots as well as rhizomes, which are a modified stem structure that you would find underground. And they have leaves as well, which in the case of ferns, we're going to refer to as fronds. Fern frond is a leaf. Coiled young fronds, like this one in the background here, have special names as well. So we'll often refer to them as fiddleheads. So a fiddlehead fern is not a type of fern, it's not a species of a fern. It's telling you the life stage of that fern, that fern frond is young and growing and unfurling. There's some other terms for the same structure. Crozier, I believe, is named after a specific type of staff with kind of a curl at the top, or we could call it circinate vernation. Anytime you see this word vern, it means like young. So circinate is telling us that it's like a circle as the young frond is expanding. But these are all equivalent terms referring to this little spiral structure as the fern unfurls. We'll go over sort of diagram of the parts of a fern specifically. So some of these structures are the same as when we're talking about our leaves more generally. In the case of ferns, we're occasionally going to have more kind of specified words for things. Okay. So the whole thing from the base to the very, very tip of the fern is called the frond. So it includes both the blade and the stalk here. So this stalk is kind of like the stem, the stipe of the fern. Then our blade is the whole leafy part. Right, so our blade is here. Fern fronds are usually compound leaves. So that is one leaf, that is one leaf blade. But as you can see, it's separated out into leaflets, right? So we see these opposite leaflets. When we're talking about a fern, they'll often call these opposite leaflets pinna. Pinna, whenever we see pinna or pinnate, it's telling you it's something that looks like a feather. That's what that means. So these little leaflets look kind of like feathers, so they're called pinna. occasionally we'll also see, if we were to zoom in on one of these pinna, we'll see doubly compound structures. So just a kind of reminder of what I'm pulling out here, right? So say these are our pinna, right? So when we say something is doubly compound, this is compound because they're separated, attaching to that central axis and the radius. So doubly compound means we have it happening again within each leaflet, so like that. So I know for a fact that we have an example of a doubly compound fern frond in the greenhouse upstairs. So if we had one of these doubly compound structures, we would see that structure within the pinule. So the pinule is like the leaflet on the leaflet. So it would have additional looks. Here we see the roots, the tendril-like structures. And here we see the rhizome, which is below the stalk, the modified stem structure, but it's going to look like it's part of the roots. But the roots themselves are going to be kind of tendril-like. The rhizome is kind of like a nodule, kind of easier looking. Here we see a young frond unfurling. So this is our fiddlehead, and it would slowly unfurl to become a big frond like that image. So these are some of the things that we'll focus on in that one today. Well, some other structures we want to pay attention to when we're looking at ferns. Some of these structures that we'll usually see on the underside of one of the fronds. So the reproductive structures of the ferns, kind of weird looking. They have some different named parts, and we're often going to see them on the underside of a leaf, and they'll have different named components depending on our species. So what we want to talk about right now, something called the indusium. So the indusium is a sort of scale-like covering that's going to protect the sorbus and its sporangia, which are going to be the reproductive structures that make the spore. So when we say that, I'll do that later. So the indusium here would be this white sort of cap on these sporangia, which together are called a sorbus. We may also sometimes refer to big leaves and little leaves as megaphylls and microphylls. And sometimes we will say we have a megasporophyll, which means we have a big leaf, and we're saying it has spores or sporangia on the underside. Or we might say we have a little leaf with spores, which would be a microsporophyll. That's all that means. We're saying how big is the leaf, and here we're saying that it has spores on it. Because we will see that some fronds, some fern leaves are also sterile, so they don't have these reproductive structures on them. So that's why we would specify that here. So here's another image from your text showing some of these structures. So here we see our fern frond, this whole leafy part. It has a stipe or a stalk, right? So that's our stipe or a stalk. In this case, this one specifically has some scales on it. So those aren't part of the leaf coming off, but we can see there are some little afros. Here's our rhizome-like structure, the base, with the roots coming off of it. And we've also indicated the rachis, that central axis, that sort of central part of our fern frond, that point where all our little leaflets of the pinna are attaching. On the other hand, sometimes when we look at ferns, we're going to see that there are two separate kinds of structures coming out of the fern. And you've probably seen these out in the woods before. So sometimes we're going to have a sterile frond like this, and we're going to see a separate frond that's going to look very different. It's going to look like it's a bunch of seeds, basically. They're not seeds because these are seedless plants, but they are reproductive structures. So the fertile frond would be containing all these spore structures at the end. So this would still count as a frond, but now we have the two types, the sterile frond and the fertile frond. Otherwise, we're naming the structures the same. So the site is like a stalk structure. And here we see a crozier or fiddlehead here. We might refer to this as dimorphic. Di means two, morph means like a shape. So all that's saying is we have two different kind of shapes of fronds within this structure. In this image, we also have some examples of indusia. Okay, so here we see an example, much like the background of that previous picture. So we have the indusium kind of in the center, spreading over these sori here. A sorus being a sort of circle collection of sporangia. Here we see another indusium. We can see it has a slightly different shape. They're kind of out laterally on the side, but we still have an actual scale covering over our sori, covering over those sporangia. Some ferns don't have an indusium. So here we see nothing covering over these sporangia. So the sporangia have a little circle. The sorus is kind of a big collection of circles. These are just out there touching the environment. And then our final option is something called a false indusium. So in this case, we are protecting those spores. We are protecting the sporangia, but instead of creating a new growth, instead of creating kind of a scale, we just have the edge of the leaf folding over. So this is the folded over edge of one of our leaves. So we are going to talk a little bit about the life cycle of a fern, which will vary some from group to group. So we're going to go over kind of a general case in lecture today. But some things to know in general about plants and plant reproduction and plant genetics is that they get kind of crazy. So actually the species in the world that has the record for the most number of chromosomes is a fern. I mean, among animals, plants, everything. Some estimates for this fern, Ophioglossum reticulatum, are up to 1,440 chromosomes for this fern. Which you can see why the geneticists trying to make these phylogenies maybe have some trouble. Another thing that is true about plants generally, other than that, the chromosome number from any species can differ a lot. One of the chromosome numbers in your book is one fern has 11 chromosomes. But in some plants, we can have polyploidy, which means that instead of having, like a human does, 2 sets of chromosomes, plants can have more than that. They can have more than 2 complete sets of chromosomes, which makes things a little difficult. So, when we think about the basic life cycle of ferns, we're going to notice that they have sort of 2 parts to the cycle. They have alternating generations. So they're going to have different kind of reproductive structures involved in their first generation versus in their second generation. So first, we're going to create our spores. So we call this the sporophyte generation. Okay. So the sporophyte generation involves a sorus. So that was that circle of sporangia, right? So those were the structures that looked like that, right? So these individual parts are the sporangia, and the whole big thing is a sorus. They're going to create these sporangia, which are then going to create the spores. So the spores are going to germinate into a haploid gametophyte. So haploid, hopefully you guys vaguely remember meiosis, right? That haploid means we have half of the number of chromosomes if we were thinking about a typical individual as having 2 full sets. So then next we have the gametophyte generation. And in the gametophyte generation, this is where we're going to see both male and female structures. So spores aren't going to have male and female, but in the gametophyte generation, we are going to have male and female. So we're going to see two structures, a male antheridium. Antheridium is our male gametophyte structure. This is going to contain the sperm. The female archegonium is going to contain an egg. These are then going to combine to create an embryo, which is going to grow into a young sporophyte. So then the cycle will continue because we're jumping back to the top here, right? So then we're going to make our spores and then enter the gametophyte generation again. I like animation, so we're going to watch this happen. Don't worry about the extra vocabulary terms that are going to appear in this. I just think this is going to be slightly more digestible than the diagram we're going to go over in a second. That this is the least weird music I found in a fern video.
Since the earliest of times, ferns have been appreciated for their beauty and have provided shelter and sometimes food. The birth of their young coiled fronds is often been used in artworks such as the coraux. Ferns are popular in our parks and gardens, and even brought indoors to enhance our living spaces. There are as many as 12,000 species of ferns throughout the world. Some are the size of your fingernail, while others are as tall as a tree. In the wild, ferns are successfully reproducing and spreading throughout the forest. They do this by producing spores. The familiar fern is known as a sporophyte. If a fern frond is turned over, small circular areas may be seen. Each of these areas is called a sorus. In some ferns, the sorus is unprotected. In many others, it is covered by a cap, supported by a central stalk. This protective covering is called an indusium. In some ferns, this covering is cup-shaped. The sorus itself contains numerous sac-like structures. Each of these is called a sporangium. The outer wall of the sporangium consists of a layer of protective jacket cells. As the sporangium matures, a row of jacket cells enlarge to form a band known as the annulus. The outer wall of each annular cell is very thin and delicate. On the opposite side of the sporangium, several delicate root cells form. Below the jacket, there are two layers of cells called the tapetum, which nourish the fertile tissue within them. The fertile tissue consists of sporocyte cells. Each sporocyte cell is diploid, containing two sets of chromosomes, one from each parent. As each cell matures, its nucleus divides twice by the process of meiosis. Each daughter nucleus now contains one set of chromosomes. The cytoplasm of the sporocyte undergoes cleavage. This results in a cluster of four adhering cells, called the tetrad. The tapetum now begins to break down, depositing a very tough protective coat of sporopollen. Surrounded by this thick resistant wall, each cell is now called a spore. The sporangia are now ready to open. Opening is accomplished by the annulus that encircles the sporangia. The annulus begins to dry out as water evaporates from its surface. The tension or pull between the remaining water molecules and the wall now increases. The thick inner and side walls resist this. The thin outer walls are easily pulled inward. Pulling in the outer walls causes the annulus to contract much like an accordion. Shortening the annulus tears the red cells apart. As the annulus continues to shorten, the tear enlarges and the spore case opens further. When too much water is lost, the water molecules are no longer able to hold together. When this happens, the pull on the wall is released, and the annulus springs forward, closing the sporangium so fast that the spores are thrown out. This process is repeated in thousands of sporangia on a leaf, so that large quantities of spores are released. When spores land on a moist surface, those which germinate first will form bisexual or hermaphroditic individuals. A rhizoid emerges and attaches the spore to the soil. This is followed by a sheet of cells, which is the young gametophyte or prothallus. A notch forms on the prothallus that contains dividing cells. This is called the notch meristeme. Continued growth results in a heart-shaped bisexual gametophyte. The lower surface is firmly attached to the soil by numerous rhizoids. Male gametangia, called antheridia, are formed at the posterior end of the gametophyte. The outer wall of antheridium consists of green cells and a cap cell, which surround fertile tissue. At the anterior end of the prothallus, close to the notch, there are female gametangia called archegonia. An archegonia consists of a neck containing a neck canal cell. At the base of the neck, there is a swollen region called the venter that contains an egg cell. Gametangia of both sexes may be present at the same time or at different times. The timing of their appearance will determine if there will be self-fertilization or cross-fertilization. In some ferns, archegonia are formed first, and when flooded with water, a hormone called antheridiogen is released. This hormone will stimulate adjacent plants to stop growing and form antheridia, but no archegonia. A bisexual gametophyte in a female phase may therefore be surrounded by several male gametophytes, increasing the chances of cross-fertilization. Being close to the soil, The small gametophytes are easily flooded when it rains. This water plays an important role in fertilization. Water stimulates the cap cell of the antheridium to open, releasing the sperm cells. The flagellated sperm are now able to move within the water in search of an egg. Sperm consists of a spiral cell body bearing numerous flagella that move it forward. Water also stimulates the archegonium to open. The contents of the neck canal then diffuse into the surrounding water, where they act as a sperm attractant. The attractant stimulates the sperm to swim towards the open archegonium. It then swims into the opening and moves up the neck canal towards the egg cell. Fertilization is accomplished when the egg and sperm nuclei fuse. This creates a single diploid cell, the zygote. The fertilized egg remains attached to the gametophyte. Inside the archegonium on the lower surface of the gametophyte, the zygote begins to divide, forming the embryo's sporophyte plant. The uppermost part of the embryo, the foot, absorbs nutrients from the surrounding gametophyte tissue. As the embryo grows, the venter tissue stretches and eventually ruptures. A root now grows into the soil for support and to absorb water. Other parts of the embryo form the first leaf and the future stem, the rhizome. The leaf grows through the notch in the gametophyte to reach the sunlight. Eventually, the rhizome emerges and provides additional leaves and roots. In this way, a new plant is formed that matures into another spore-producing plant, completing the reproductive cycle. We have seen the fern reproductive cycle contains two plants, a large familiar spore plant or sporophyte, and a tiny gamete plant or gametophyte. Using spores to travel great distances and gametes which introduce genetic variability, ferns have spread and evolved throughout the world.
So we saw there that our main structure that we see that we think of as the fern is that sporophyte generation. We have this tiny gametophyte generation, which is where we have the egg and sperm cells. So we can look at this also using a diagram from our textbook, but I prefer the movement. But here we can see that we're seeing the same thing. So we're going to start. We'll start with the adult sporophyte. So if we're looking at an adult sporophyte, so we're looking at a fern frond, you're going to see on the underside of the frond, the sori. So the sorus is that collection of sporangia, or our spore-producing structures, potentially covered up by an inducium. So here we see that zoomed in. So we're zooming in on one of those sori. So here's our covering, the inducium. And here are our sporangia, a collection of which is called our sorus. If we zoom in on one single sporangium, you see that this is the location where our cells are undergoing meiosis, forming our spores. And they're in this, I don't even know how to describe this structure. This is a circular structure that produces like a mouth to throw our haploid spores about the environment. We need moisture changes in order to actually send these spores into the environment, because this is what causes the sporangia to burst open as the structure, the annulus, shrinks. And they literally refer to ferns as throwing spores. That's why we were showing that flinging kind of movement. They actually do that. So the spore begins to then germinate as a sort of intermediate structure called the prothallus, which is turning into our mature gametophyte, which is kind of heart-shaped or saddle-shaped. So on the gametophyte, depending on the fern we're talking about, we may have both male and female structures, or we may have those on separate gametophytes, but we can zoom into them on the same image anyway. So here we see our archegonia producing the egg cells. And on the opposite side, these tendril-like structures, those are our male structures, the antheridia, producing the sperm cells, which have flagella. So now we are interested in another moisture change in order to combine our sperm cells and our egg cells. When we combine them, they become a fertilized zygote that creates a young sporophyte growing out of the gametophyte structure, which then degenerates as the sporophyte grows, and then we are back to the adult. So that is our basic fern life cycle. But depending on the specific structures of a specific species, we may have a little bit of variation here. Yes, before we go to lab on Thursday, I want to take a look at a couple of examples of these groups, these four main divisions that we're interested in. So this is an image of a club moss, one of our lycophytes. So that most basal group, that first one to branch off. So our lycophytes have really small, simple leaves. You'll see that these really don't look like our fronds, right? These look really different than our typical fern frond. When we see that typical fern frond, really we're mostly talking about the tree ferns when we're talking about those structures. So it has these small, simple leaves. Since they're small, we might call them microphyls. They're usually just going to have one vein in the center. They're going to be spiraled around, radius spiraled around the stalk. So we might call this a rolled effect. Modern lycophytes are really small, but some ancient examples that they have, some fossils, are huge and tree-like. But the ones remaining are almost moss-like in structure, which is why many of them are referred to as club mosses. So they're not actually moss, they're actually in the fern allies group, but because of that small structure, they get confused a lot. Next, we have our whisk ferns and Silotophyta. So these are primitive looking in that they just often look kind of like sticks, basically, right? So we see these scaled kind of stem-like outgrowths. And we can see the sporangia located along them, so they do still have spores. That's what unites this group of the ferns and the fern allies. But they don't really have much of a leaf structure. So they lack true leaves. They actually also lack roots. So they're going to look something like this. Whisk ferns. Next, we have our horsetails. I have these around here, and they honestly look like zombie plants when they come up in the spring. We'll probably see some popping up around campus in a month or two. Our horsetails, like Microphyllophyta, there's actually only one family and one genus remaining today, the Zeotaceae, which Equus is horse. But this was a group that included more individuals in the past. Structures that we see on a horsetail are astrobolas. So that basically refers to a cone, but this isn't going to be like a woody cone like a pine cone. It's kind of a spongy, fleshy cone. So this is where we're going to see the sporangia in the horsetails. And when we look at the stems, they're going to be hollow and they're going to have these kind of joints. And when we see branches or leaves coming off, they're going to come off those joints, those nodes in a whorled arrangement. So we'll see examples of these in the future. And then we have our true ferns, which are going to be characterized by large fronds with branching vein networks. So more than one little vein in the center. We'll have multiple different types and multiple different examples. In this case, we're seeing one where we have both the sterile and fertile frond structures. So you can see that. Okay. So here on the left side, it's referring to questions 60 to 68 in the textbook. So you can look over these as you're studying for your quiz next week. I'll put one of these on the quiz, so this will count as studying for the quiz. I'm not going to go over these right this second. So my final thing for you for today is about that phylogeny practice. Did everybody get a chance to look at it? I know I put up a key, but I want to know if people want to go through additional explanations or want to talk through any parts of that. I tried to put the explanations as detailed as I could in that key, but I don't know how close I got. Anybody have anything from that they want to talk about? It worked. Okay. Okay. In that case, we're just going to have a really short day today. We'll do a lab on ferns on Thursday, and we'll find another day with slightly nicer weather where I don't know if it's actually snowing or not right now, but a better day where we can go out and see some actual examples of trees. All right. That closes us out for now.